Thank you for the introduction, and I'm very happy to be here today. I would like to take the next 10 minutes to talk about HyGIeaCare's non-invasive patented method to obtain unique intercolonic microbiome. HyGIeaCare was established in 2015 and currently operates 6 centers in the US and building 4 additional ones. In the heart of our operation is our FDA cleared system that also has CE mark that allows us to offer 3 services. One is the HyGIePrep, which is a better way for colonoscopy prep. The other one is the HyGIeRelief, which is an unmedicated constipation relief. And the third is HyGIeBiome, which is what I'm going to talk about today. So we are all here because we know what is the potential of the microbiome. We know that the majority of known disease can be associated and or reflected in the composition and function of the gut microbiome. We also, in HyGIeaCare, we partner with gastroenterologists and set centers at point of care and have access to colorectal cancer screening population, which allows us to collect samples from all prevalent disease in the population. We also know that different organisms live in different environments, right? The way giraffes and bears do not share habitat, also microbes like their own habitats and ecological niche. Now, the literature is filled with indications that the colon has its own biogeography, which makes sense because the colon starts at the cecum with the drip from the small intestine and runs for 6 feet until stool is being secreted from the rectum. So we at HyGIeaCare believe that it's really important to sample biogeographically relevant samples from the gut, as Denise Kelly indicated this morning. So the HyGIeSample. The HyGIeSample is our unique and patented method to obtain non-invasively intercolonic microbiome. It also enables a collection of microbiome without the ill effect of an oral purgative, which is usually provided to patients before their colonoscopies. So 2 years ago, we decided that we want to test our HyGIeSample hypothesis. The first one is that HyGIeSample is profoundly different than stool sample as per the microbiome. And the second hypothesis was that the HyGIeSample is complementary to stool sample for biomarker and drug design, biomarker discovery, drug design, and also patient stratification. So we ran, to test this hypothesis, we ran a prospective study with the goal of identifying the genomic related differences between HyGIeSample and stool sample. We enrolled up to 20 healthy HyGIeaCare screening population patients, provided them with a home collection kit to collect their stool, and during their HyGIePrep, we collected HyGIeSample from the beginning, middle, and end of their process, which should provide us with the entire biogeography of their colon from left transverse and right colon. We did analysis for 14 sRNA metagenomics, and we used it for taxonomic and phylogenetic analysis, and also to recover biosynthetic gene clusters to look at functions. And to cut a very long story short, we found major differences between HyGIeSample and stool sample, and I'm going to present a few of our results now. So more microbial strains were found in HyGIeSample, and 31% of these detected strains were unique to HyGIeSample. Also, when we're looking at the phyla level, when we focus on the top 4 phyla, we can see a significant increase in Proteobacteria and decrease in Firmicutes. When we throw in additional 3 phyla and look at the top 7 abundant phyla, we can see sample type specific microbiome landscape. Looking at the ratios between key phyla for when we look at the F2B or P2A, we can see differences between the HyGIeSample effluent and the stool. Looking at the genera level, we can again see differences in the abundance of genera across different phyla. The P2B ratio is a known marker for differences in diet and lifestyles. Again, we see significant difference between stool and HyGIe sample. And in the species level, we see changes in species abundance that can, of course, be translated to differences in metabolic network. We also heard during this conference talks about key species and about Akkermansia, and we can definitely see that we detect significant difference between species abundance in stool sample and in HyGIe sample. We also can look at the biogeographic biodiversity, where we found profound changes in the microbial ecology of the gut versus stool samples. You can see alpha diversity, you can see beta diversity differences. You can also see that when we compare sample effluent versus stool from a differential abundance perspective, where you can see from left to right, effluent 1, 2, and 3 is left colon transverse and right going towards the small intestine. The closer we get to the small intestine, the higher the differential abundance versus stool is. From perspective of biosynthetic gene clusters, 69% of the detected BGCs are unique to HyGIe sample, which infers a huge metabolic potential. You can see it here. You can also see that this also have a biodiversity aspect where the different segments of the colon provide different metabolic networks. So to conclude the HyGIe sample, the HyGIe sample reflects the biogeography of the colon, including the ascending colon, the right colon, which is the closest we can non-invasively get to the right colon, to the small intestine. HyGIe sample is also complementary to stool sample when you look at the microbiome. It's the only patented non-invasive way to obtain intercolonic microbiome. We at HyGIeCare believe the HyGIe sample is going to improve significantly the probability of success in clinical trials, including patient stratification, biomarker discovery, and drug design. Yes. HyGIeBiome, which is the research arm of HyGIeCare, offers a new way to look at the microbiome. It is uniquely poised to support gut microbiome studies based on 3 significant differentiators. One is the HyGIe sample management and collection. The second one is our access to patients with a wide range of prevalent disease at point of care. And 3 is consented access to patients' electronic medical records. So when you choose to run your studies with HyGIeCare, you basically get intercolonic relevant, biogeographic relevant samples that are sampled by trained personnel in a controlled environment. And because of our partnership with the gastroenterologist, you actually get access to population, ease of enrollment, and seamless access to their EMR. Currently, we are looking at a few indications that are basically derived from 2 of our biggest, or 2 of our main differentiators. One is our ability to get samples from the colon, including the right colon, where we look at IBD and pancreatic cancer. I don't think that we need to go in length about the importance and the unmet need in IBD studies. With pancreatic cancer, again, we don't need to talk about the need, but it's really important to say that because we can get the drip from the small intestine, we are highly likely, much more than stool sample, to actually find a biomarker or some kind of indication for pancreatic cancer. The other factor is actually derived from our partnership with the gastroenterologist and us being at point of care, where we have access to patients that are being screened for colorectal cancer. We had more than 18,000 patients so far, and our access to constipated patients where they can very rarely pass stool, and it's really hard to do stool-based studies with them. Also, we have a lot of returning patients, so we are poised really well to do longitudinal study with them, also with the colorectal cancers. So with that, I wanted to thank you for your attention and invite you to join us and leverage on our advantages for successful research outcome. Thank you.
Great, Oli, thank you so much for that presentation.